Modern Swedes are the descendants of Vikings. Everyone knows this. It's in the textbooks. It's in the DNA testing commercials. It's in the way the word Nordic gets used as if it means something ancient and unbroken and continuous. The science confirmed it years ago. Case closed. Except, in January 2023, a team at Stockholm University published the largest ancient genomic study ever conducted on Scandinavia. 297 ancient individuals, 16,638 living Scandinavians. 2 columns of data that should have matched each other. They didn't. What Dr. Ricardo Rodriguez Varela and his colleagues found was this. In multiple regions of Sweden, the people buried there during the Viking Age carried more non-local ancestry than the people living there today. Read that again. The skeletons are more diverse than the living. The past is more complex than the present. The heirs, genetically, are smaller than the ancestors. This isn't a story about what Vikings brought home from their raids and their trading routes and their settlements. That story is real, but it's the wrong story. This is the story about what happened after, about what Sweden lost, about a deletion that left no perpetrator, no record, and almost no trace, except in the data if you know exactly where to look. But here's where it gets strange. The deletion didn't start in the Viking Age. It started before Sweden was Sweden, before Scandinavia was Scandinavia, before anyone on that peninsula had a name for themselves at all. If you've ever wondered where you actually come from, not the family story, not the DNA test percentage, but the real, layered, uncomfortable biological answer, then subscribe now, because what comes next goes deeper than almost anyone is willing to follow. The piece of data sitting in that Stockholm study is missing a variable. 3 genetic sources entered Scandinavia during the Viking Age. The study names all 3. It cannot name the force that removed them. That variable is still missing from the scientific record, and it may be the most important discovery still waiting to be made. What's missing from the record goes back much further than the Vikings. 9,880 years further, to be precise. There is a site in western Sweden called Husby Clev, and in a layer of earth dated to approximately 9,880 years before the present, archaeologists found something unexpected. Not a body, not a weapon. A piece of chewed birch bark pitch, the Mesolithic equivalent of adhesive, used to fix a stone blade to a handle. Small, dark, hardened. And inside it, preserved by the chemistry of organic tar over nearly 10 millennia, human DNA, not just traces, genome-wide data, from 3 individuals who left their genetic signature in the act of chewing gum. Dr. Natalaja Kashuba at Uppsala University and Dr. Anders Getherström at Stockholm University analyzed those 3 individuals in a study published in 2019. What they found was that these people belonged to the Scandinavian hunter-gatherer group. The first confirmed humans to colonize Sweden after the glaciers retreated north. Their Y chromosome haplogroup, the paternal lineage passed from father to son, I2, carried near exclusively by every male in Mesolithic Scandinavia. That piece of birch bark pitch is older than the pyramids at Giza by 6,000 years. The person who chewed it had no name for Sweden, no concept of Europe, no language we could read. And yet their genome is the oldest layer under everything that came after, the bedrock. This is what recognition feels like in ancient DNA research. You look at a number, less than 3%, and you understand what it means before you can articulate it. Less than 3%. That's the frequency of Y haplogroup I2 among modern Swedish and Danish men. The genetic signature of the people who lived in Sweden for 5,000 years, before farming, before metals, before anything we'd call civilization, survives today in fewer than 3 out of every 100 men in the country those people built. The first Swedes were nearly erased before a single longship was ever constructed. One of the three Husby-Clev individuals yielded genome coverage of 44 times, among the best-covered Mesolithic human genomes ever sequenced anywhere. Their oral microbiome was also preserved. The metagenomic analysis, published in 2024, found evidence of dental disease. They suffered. They had specific oral health problems. They were not romantic archetypes or noble primitives. They were people with bodies that hurt in specific, documentable ways. And then, around 5,900 years ago, other people arrived. And the I2 signal began, very slowly at first, then faster, to disappear. But here's what the data shows that the simple replacement story misses. They didn't vanish immediately. Something more complicated happened first. The I2 haplogroup of the Mesolithic Swedes should have disappeared cleanly. A new population arrives from Anatolia, farmers, carrying almost no hunter-gatherer DNA, and the original inhabitants get absorbed or displaced. That's the simple version. The data is not simple. For centuries after, Anatolian Neolithic farmers established themselves in the interior of Scandinavia, building their megalithic tombs, growing their grain, raising their cattle. There were still pockets of hunter-gatherers on the coasts, on the islands, on the fringes. The pitted ware culture of Gotland and the Swedish coastline retained substantial hunter-gatherer ancestry well into the Middle Neolithic. Dr. Pontus Skoglund at the Francis Crick Institute, in research that anchors much of what we now know about early Scandinavian genomics, showed that these two populations shared a shoreline for centuries. Different pottery, different burial practices, different diets confirmed by isotopic analysis, and no confirmed archaeological evidence of war between them. Think about what that means. Two genetically distinct populations on the same coastline for hundreds of years. Not mixing, not fighting, parallel. There's a burial on Gotland from approximately 3000 BC. A young adult female, pitted ware culture. Isotopic analysis, predominantly marine diet. Hunter-gatherer ancestry dominant in her genome. 40 kilometers inland from her burial. A funnel beaker tomb. Anatolian farmer ancestry. Terrestrial diet, completely different material culture. Two populations on one island living parallel lives inside parallel genomes. The coexistence looks peaceful. The genomic outcome does not, and yet, the data said the exact opposite of assimilation. The hunter-gatherer I2 lineage persisted in pockets even as farmers surrounded them, until something changed that was not the farmers. Around 900 BC, a signal appears in the Scandinavian genomic record that has nothing to do with Anatolian farmers and nothing to do with Mesolithic hunters. It comes from the east, and when it arrives, the I2 haplogroup stops. Not slowly, not gradually. It stops. But here's the part that hasn't been fully resolved. Some researchers point to traces of Western hunter-gatherer ancestry in modern Scandinavians that exceed what the Neolithic replacement fully accounts for. The signal is small, but it's there. Somewhere in Sweden tonight, a person may be carrying the direct male lineage, Y chromosome haplogroup I2, of the Husby-Clev birchbark chewer, in a direct, unbroken patrilineal chain across 10,000 years. They almost certainly don't know it. Around 3100 BC, somewhere on the Pontic-Caspian steppe, a grassland corridor, stretching from the Danube Delta to the foothills of the Ural Mountains. A mobile population was moving. They had horses, they had wagons, they had cattle. And they had a genetic profile that researchers would later call Western steppe herder ancestry, a combination of Eastern hunter-gatherers and Caucasian hunter-gatherers that had been mixing on the steppe since approximately 5000 BC. These people were about to become the single largest source of DNA in the modern Swedish genome, and they would do it in a geological instant. The Scandinavian battle-axe culture, the local expression of the broader corded ware horizon, entered Sweden around 2900 BC. Research from Dr. Anders Gutherström's group and associated studies, published in the Proceedings of the Royal Society B, showed that battle axe culture individuals carried approximately 75% Western steppe herder ancestry. 75%. In a population that replaced, within a few centuries, the genetic landscape that had existed for 5,000 years. Why haplogroup R1a M417, absent from every Scandinavian male skeleton before 900 BC, appears with the battle axe culture and becomes one of the defining paternal lineages of modern Scandinavia. The As2 lineages of the Mesolithic? Gone. The farmers who preceded the steppe migrants? Reduced to a fraction of their previous frequency. The specific Scandinavian subclade that emerged from this, R1AZ284, is still carried by millions of people in Scandinavia today. Stay with me, because this next part is the reason this video exists. If you are of Swedish or Scandinavian ancestry, if your grandmother came from Stockholm or Uppsala or anywhere on that peninsula, the single largest genetic component in your body right now did not originate in Scandinavia. It originated on the Eurasian steppe, carried thousands of miles west by people who probably spoke a Proto-Indo-European language, left no written record of any kind, and entered a country that had no name for itself. They are not in the history books. They are in you. There is a battle axe culture burial from central Sweden, approximately 2800 BC. Adult male, tall and robustly built, confirmed by a skeletal measurement. Haplogroup RZN284, buried with a battle axe. The axe is a cultural marker. The genome is the real legacy. It is still replicating in Stockholm today, nearly 5,000 years after that burial mound was sealed. And yet, this steppe migration, whose genetic impact on Sweden rivals any event in the country's documented history, came from a source population that is still debated. The Yamnaya culture of the Pontic steppe is often named, but the specific people who carried R1a M417 into Scandinavia were not straightforwardly Yamnaya. They moved across a continent. They reshaped an entire peninsula's genome, and they left no name. The people who built the genomic foundation of modern Sweden are, to history, completely anonymous. Actually, hold on. Before we go further, I want to ask you something directly. When you think about your own ancestry, wherever you're from, do you think about it as something fixed? Something that goes back in a straight line? Or do you already suspect it's more layered than that? Drop it in the comments. Because what I'm about to show you about Sweden is going to land very differently depending on your answer. The steppe migrants became Scandinavian. Over 2,000 years, the genome settled. The Bronze Age Nordic profile emerged, a specific mixture of steppe ancestry, Anatolian farmer ancestry, and residual Western hunter-gatherer components, and by the Iron Age, it was stable enough to be identified in the archaeological record as distinctly Scandinavian. Then, starting around 750 CE, everything in the genomic record shifts again. There is a cemetery at Birka, a Viking-era trading town on an island in Lake Mälaren, west of Stockholm. The burials there have been analyzed extensively. One in particular became famous, a warrior burial confirmed female by osteological analysis, interred with weapons, a gaming set, and 2 horses. Her genome, predominantly Scandinavian ancestry, with Eastern European contributions. She is not the surprise. By 850 CE at Birka, the surprise is that almost every burial contains something different. British-Irish ancestry in one grave, Eastern Baltic ancestry in the next, Southern European contributions in another. The Trading Post Cemetery is a map of the known world, compressed into one island. The Cell 2023 study put numbers on this. British-Irish ancestry was widespread across all of Scandinavia from the Viking period. Eastern Baltic ancestry concentrated specifically in Gotland and central Sweden. Southern European ancestry entered more strongly in the south. 3 distinct genetic flows, arriving simultaneously within one 300-year window. Dr. Ricardo Rodriguez Varela and Dr. Agnar Helgason at Decode Genetics in Iceland were explicit. Viking Age Scandinavia was not a genetically homogeneous Nordic population exporting itself to the world. It was the world arriving in Scandinavia and leaving a genomic mark. This isn't just ancient history. This is in your DNA right now. If you've taken a commercial DNA test that returned a percentage labeled Scandinavian, that number was built from a dataset averaging across 8,000 years of replacement, arrival, and deletion. The profile the algorithm calls Nordic was assembled from at least 5 distinct population layers. 2 of those layers are poorly understood. One of them, the most consequential one, has not yet been fully explained. In the large-scale Viking genomic study published in Nature in 2020, researchers found something that made the Birka Cemetery look almost ordinary. A Viking expedition to Greenland contained individuals confirmed by DNA to be closely related, brothers most likely, traveling together to the edge of the known world. Their lineage ends there. The colony failed. Their genomes stop at a latitude no one in their family had ever reached before. And in that stopping, there's something that feels important and cannot quite be named. The genetic profile be sold as Nordic heritage. The one in the test kit, the one in the documentary thumbnail, is partly a post-Viking construction. It was assembled after the diversity of the Viking Age was removed, which means we have been mistaking the aftermath for the original. 2 civilizations, the diverse, cosmopolitan Viking world and the more genetically uniform modern Scandinavian population, are being sold as the same thing. The data says otherwise. There is a town called Sigtuna, north of Stockholm, one of the oldest towns in Sweden. Around 1100 CE, 50 years after the end of the Viking Age, it was a functioning cosmopolitan settlement. Its streets held merchants whose skeletal isotopes suggested origin in what is now Poland or Lithuania, clerics whose genomic profiles showed British-Irish contributions, craftsmen from the Eastern Baltic. Genetically, this was one of the most complex human settlements in Scandinavian prehistory. And across the next 300 years, quietly, without a single documented event explaining it, this complexity contracted. The Cell 2023 study made this explicit. Dr. Gutherstrom's team stated it directly. In several Scandinavian regions, current levels of external ancestry are lower than those found in Viking-era and medieval-era burials from the same geographic locations, the same soil, the same towns. Less diversity in the living than in the buried. And here's what no one has been able to confirm. Why? The researchers offer at least 2 explanations, neither confirmed. First, Viking-era immigrants may have had fewer children than the pre-existing local population. Second, the Eastern Baltic gene flow may have been predominantly female, women entering the genetic record through mitochondrial DNA, but leaving weaker autosomal signatures across generations. Both are genomically consistent with the data. Neither explains it fully. Everything I just told you, hold on to it, because it's about to mean something completely different. The Black Death reached Sweden in 1350 CE. It killed approximately 1/3 of the population in roughly 12 months. That is a documented catastrophe. And the tegenomic contraction, the disappearance of Viking-era diversity from the modern Swedish gene pool, overlaps with that timeline. If Viking-era immigrants and their descendants were disproportionately concentrated in urban trading centers, which the isotopic and archaeological evidence suggests they were, then they died in disproportionate numbers when a disease that moved fastest through densely populated areas swept the continent. This has not been confirmed by the current evidence, but the correlation sits in the data like a question that has not yet been asked in the right way. A medieval burial from Sigtuna, 12th century. Adult male. Eastern Baltic ancestry confirmed by genome. Buried with Christian rites. a merchant scale found near the body. His direct male line descendants cannot be traced forward to the modern Swedish genomic database. He left no confirmed echo in the living. He arrived, he traded, he died. He was buried with the correct religious rites of his adopted home. And then, somewhere between his death and the present, his genomic line went quiet. Modern Sweden carries a north-south genetic divide. Higher Uralic ancestry in the north, higher steppe-derived and Anatolian farmer ancestry in the south. The Cell 2023 study found this cline existed in the Viking Age, and possibly even earlier. The genomic fault line running through Sweden today is older than Sweden as a political entity. It was not created by modernity. It was inherited from a time before anyone there called themselves Swedish. The unfinished war, the genetic tension between North and South, has been running for at least 1,200 years, possibly longer, and it is still running now. The North-South division in Scandinavia has a name in the genomic literature. It is driven primarily by Uralic ancestry. The genetic signature associated with Uralic-speaking populations across a corridor stretching from Siberia through Finland into northern Scandinavia. The Cell 2023 study found Viking-era burials in northern Norway carrying the Sami characteristic mitochondrial haplogroup U5b1b1a. The people we now call the Sami, or their genomic predecessors, were already in the north during the Viking Age. The cline was already drawn. Dr. Matthias Jakobsen at Uppsala University and Dr. Kari Svanson at Genetics traced the Y chromosome haplogroup N1A1, associated with Uralic-speaking populations, into Viking-era and medieval burials in northern Scandinavia. The same corridor that connects modern Finns, modern Sami, and Siberian populations to the east left its mark on northern Sweden 1,000 years ago. The genetic north-south difference in modern Sweden is not a recent construction. It's ancient, continuous, and stable across at least 12 centuries of documentation. So, we have the full picture now. Mesolithic hunter-gatherers, I2 haplogroup 10, season years of occupation, now under 3%. Anatolian Neolithic farmers, funnel beaker and TRB cultures, megalithic tombs, coastal coexistence with hunters. Pontic steppe migrants, battle axe culture, 75% western steppe herder ancestry, R1AM417, near total male paternal replacement. Viking Age influx, British Irish, Eastern Baltic, Southern European, 3 simultaneous genetic flows. Uralic North, Sami ant stored N1A1 drawing the fault line that persists today. Stack those 5 layers, and you have the modern Swedish genome. The picture seems complete. The mystery appears resolved. But the Cell 2023 study contains one more number. In the regions where Viking-era external ancestry is best confirmed, where the British-Irish contributions, the Eastern Baltic markers, the Southern European components appear in multiple burials across multiple sites, modern Scandinavians in those same regions carry measurably less of that ancestry than the Viking-era skeletons beneath their feet. Not more, less. The Viking Age did not deposit its diversity permanently into the modern Swedish gene pool. It deposited it, and then something removed it. Modern Swedish identity is not the inheritance of Viking diversity. It is what remained after diversity was removed. The entire field assumed the Viking Age was a net genetic contributor to Scandinavia. The data in several regions shows the opposite. The story was backwards. The cosmopolitan world that the Norse built, the genomic complexity that made the Viking cemetery at Birka look like a map of 3 continents, was deleted, and no one can fully explain how. Last video, we followed the genetic thread into the Gullah-Geechee people of the American Southeast, a community whose DNA carried the specific signatures of West African populations that had survived the Middle Passage. What that episode and this one share is the same question. What happens to a genome when a people are moved or removed against the logic of their own survival? In both cases, the deletion left traces. In both cases, the science found them. If you watched that episode, drop in the comments what you think connects these two stories. Because the answer, I think, is more uncomfortable than either video says out loud. There is a woman, let's say she's in her 60s, living somewhere in Sweden. She's taken a commercial DNA ancestry test. The result came back: 94% Scandinavian, 4% British Irish, 2% unassigned. She shared it with her adult children. They thought it was interesting, maybe even reassuring, a number that confirms what they already believed about themselves. The number is not wrong, but it is not the full story. What that result is measuring is the current endpoint of a process that deleted several layers of what was there before. The British Irish 4% is a remnant of a signal that, in a Viking-era burial from the same geographic region, might have appeared at 15 or 20%. The 2% unassigned may contain traces of Eastern Baltic ancestry that the algorithm can't cleanly classify because the source population itself is so reduced in the modern sample set. The 94% Scandinavian is itself a compressed average of 5 distinct ancestral layers, 3 of which arrived on that peninsula from outside it. The number feels like identity. It is actually archaeology. Dr. Agnar Helgason at Decode Genetics and Dr. Gutherström's team at Stockholm University offered at least 2 genomically consistent explanations for why the Viking-era diversity contracted. Differential reproduction, the possibility that immigrant populations in Scandinavia simply had fewer surviving children than the pre-existing local population, and sex-biased migration, particularly for the Eastern Baltic gene flow, which may have entered predominantly through women, leaving a weaker autosomal signature across generations even as their mitochondrial line persisted. What they found next changed everything they thought they knew about this, because neither explanation is sufficient on its own, and sitting behind both of them is a timeline that cannot be ignored. The Black Death reached Scandinavia in 1350. It killed approximately 1/3 of the Swedish population, possibly more in the urban centers where trade happened, where merchants settled, where the descendants of Viking-era immigrants were most likely concentrated. Yersinia pestis, the bacterium that caused it, spread fastest in exactly the environments where cosmopolitan mixing produced cosmopolitan populations. The temporal overlap between the plague's arrival in Sweden and the genomic contraction visible in the data is not confirmed as causal, but it is not coincidental, and the field has not yet fully examined those medieval-era plague burial pits with the genomic tools that are now available. The merchant in Sigtuna with the Eastern Baltic genome and the merchant scale, buried in the 12th century with Christian rites, leaving no forward genomic trace in the modern database, may have descendants who died in those burial pits 200 years later. The diversity that should be in the modern Swedish genome may be concentrated in the mass graves of the 14th century, in the dead, waiting, still, to be sequenced. Here is the finding that carries the weight of the whole episode: a genetic marker associated with immune response. The ERAP2 variant studied in populations exposed to the Black Death shows evidence of strong selective pressure in exactly the period when the genomic contraction occurred. Research published by teams at the University of Chicago, analyzing ancient DNA from individuals who died before, during, and after the Black Death, showed that certain immune variants were dramatically amplified by the plague's selection pressure. The plague didn't just kill people, it filtered the genome, and if it filtered preferentially against populations with less long-term local immunity, recent arrivals, immigrant communities, then the contraction in the modern Swedish genome has a mechanism, an ugly, impersonal, microbial one, but a mechanism. This has not been confirmed in the Swedish context. The research is ongoing. which means the most important chapter of Swedish DNA history may not yet have been written. The birch bark pitch from Husby Clev is in a museum case in Stockholm. It is small, dark, and unremarkable to anyone who doesn't know what it contains. Inside it, the genome of a person who lived in Sweden 10,000 years ago, who had dental disease, who chewed adhesive to fix a stone tool, who left as their entire legacy a piece of hardened organic material, and a genome that now survives in fewer than 3 out of every 100 men in the country they inhabited for 5 millennia. This is what the genomic record of Sweden actually shows, if you follow it from the beginning without choosing where to start. 5 layers, 5 replacements, 5 moments where what was there before became, in the genomic language of the evidence, a minority. The I2 hunters, reduced to 3%. The Anatolian farmers, diluted by steppe ancestry. The steppe migrants, amalgamated into a Bronze Age Nordic profile. The Viking-era cosmopolitan influx, contracted by forces that remain incompletely named. And now, the living. The people walking the streets of Stockholm today are the survivors of all 5 of those events. They carry traces, sometimes large, sometimes almost invisible, of every wave. But they carry less of the most recent pre-modern wave than the skeletons buried in those same streets carry. And that gap, between what the dead contain and what the living have inherited, is where the real history of Sweden lives. If you are Scandinavian or of Scandinavian ancestry, consider this. The ancestor who came to your family's home region during the Viking Age, the British trader, the Eastern Baltic woman, the Southern European merchant, may have survived the raids and the winters and the social friction of arriving in a foreign place, only to leave their genome to a line that ends before it reaches you. Not because of war or expulsion or documented persecution, because of plague or differential birth rates, or a form of quiet social pressure that left no record except in the contraction of a data column. They were there. The science confirms it. And then they weren't. Subscribe to this channel. Not because we give you answers, because we don't always have them, but because the questions we're finding in this field are the kind that change how you understand what you are and where you came from. The Cell 2023 study ends with a specific statement from Dr. Rodriguez Varela that most summaries did not quote. He said the field needs more individuals from the medieval period to understand when and why the reduction in non-local ancestry occurred. More individuals from the medieval period. More data from the gap between the Viking Age and modernity. The answer to the central question of this episode is sitting in the ground beneath Swedish cities, in burial layers from the 13th and 14th centuries, waiting for genomic sequencing tools to reach them. The most important thing archaeogenetics has found about Sweden is not what the Vikings were. It is this: modern Sweden is a simplified version of what existed before it, a reduction, a contraction, a country built in part on a deletion that no one ordered and no one recorded, and that the science is only now beginning to measure. What the Swedes erased, they may not have erased on purpose at all, which somehow makes it harder to look at.